This week's Biology 1440 lab will focus on the topic of osmosis. We have two basic research questions. How will hypo and hypertonic environments affect the appearance of plant cells? And how will the mass of potato samples change in response to varying concentrations of sucrose? Our major learning goals are to conduct an investigation on diffusion using the scientific method. So you'll have to determine your controls and your various concentrations. You'll have to use the mass balance equation to make those concentrations. You will then produce a standard curve based on your data and use the data to then determine an unknown concentration of sucrose. You will observe osmosis in Elodea cells, improve microscopy skills, and be able to identify the parts of a microscope. So diffusion is basically the movement of molecules from a high concentration to a low concentration. This only makes sense if you understand that as long as there's energy around, that molecules move. So in the above diagram, there are dye, let's say food coloring molecules in some water. There's a membrane shown there. The membrane has pores in it that are small enough to allow water through, or I should say allow the dye through, and the dye molecules are moving, and they will cross that membrane. And eventually you'll end up with concentrations fairly equal on both sides of that membrane. You have dye molecules on the left, you have dye molecules on the right. Now let me point out that just because we have equal concentrations on both sides doesn't mean the dye molecules stop moving. They still cross the membrane. It's just that for every molecule that crosses over to the right, another tends to cross back over to the left. The same kind of an idea applies when you have more than one solute. Eventually you have fairly equal concentrations of each one on either side of that membrane, more or less. Okay, now we're going to add in another complexity here. Now we're going to add a membrane that has pores in it that are too small for the solute to cross. But water can still cross, but the solute cannot. So in this example, we have this U-shaped glass tube with an equal volume of solution on each side. However, on the left side, we have a low concentration of sugar, which is the green dots here. On the right side, we have a high concentration of sugar. It's better maybe to think about this in terms of water rather than sugar. On the left, we have a high concentration of water molecules that are free. And what do I mean by free? The molecules on the left side, the water molecules, are less likely to be involved in a hydration shell around these sugar molecules. But on the right, where there's more sugar, these water molecules tend to stick to that sugar. There is a higher concentration of sucrose, so therefore there is a lower concentration of free water molecules. That is water molecules that are not stuck to the sugar. Okay? Diffusion always occurs from areas of high concentration to areas of low concentration. So the left side of the membrane over here has a high concentration of water, or I should say free water molecules, while to the right there is a low concentration of free water molecules. Therefore, the water molecules can drift from the left to the right much more rapidly than they can drift from the right to the left. There are some free water molecules on the right, but nowhere near as many as on the left. So eventually you end up with an equal amount of free water molecules on each side of the membrane, and the actual volume of water will shift, and you'll actually have a much higher level of fluid on the right side of the membrane than on the left. I like to describe it as if you walked, let's say you're in the classroom and you walk out into the hallway through the door and you sit down on a horse or on an elephant. You can't get back through the door again. That's sort of what's happening to this water. It drifts over this way and then it gets stuck to something really big, and now it can't come back through that opening again. This has massive implications and consequences when it comes to biology. If you place an animal cell into what we call a hypotonic solution, this is a solution with lots of free water molecules. So it's pure water basically. In that case, water will flow into a cell rapidly, and in the case of an animal cell, it will lyse or burst the cell. In the case of a plant cell, this is normal, and plants actually thrive under these conditions. Water flows into the cell, swells the cell up, but the cell wall prevents the cell from overexpanding and bursting, and that's how plants maintain their rigidity. If you place cells in an isotonic solution is normal for animal cells. There's an equal amount of water coming in as is coming out. That's good for animal cells. For plant cells, they become flaccid, meaning like think of celery that has sat in the fridge a little too long and it's kind of limp, right? That's not good for plant cells because the central vacuole loses a bunch of fluid and it's not pushing out against the cell wall anymore, and the plant actually loses some of its structural integrity. If you put an animal cell in a hypertonic solution, that's a solution with very little free water but with a lot of dissolved solute, then water tends to leave the cell faster than it comes back in, and the cell will actually shrivel up. And in the case of a plant cell, the whole cell itself doesn't shrivel up because the cell wall doesn't shrink too much, but the cytoplasm inside dehydrates and pulls away from the cell wall, and you have what you call a plasmolyzed cell. And that cell's probably not going to recover after that. So I would advise that you refresh your memory on the parts of a scope, as we'll be using them again this week, and remind yourself how to make a wet mount because we'll be doing that as well. One rule, just to remind yourself when you're cleaning lenses, never ever use a Kim wipe or a paper towel. Always use lens paper when cleaning your microscope lenses, and always put your scope away properly with the cord tightly wrapped up around the microscope. So we're going to observe Eladia this week. I'll talk more about that in class, but it will be cool. You'll be able to see the cell walls. You'll be able to see the chloroplasts inside the cell, and we'll watch what happens when we put them in pure water versus very salty water. So what we'll do is we'll collect data, and then we'll discuss it at the end of lab, and you'll have an assignment to work on. One thing I want to point out is your assignment is going to be to make a scatterplot. I'd like you to do this on Excel. Please check out the videos on the lab website if you don't know how to do this yet. But you're going to make, this is obviously not the graph we'll be making, but it is a scatterplot, much like we'll be making. You'll insert a best fit line. You'll also tell it to insert the equation of the line. And this is what we call a standard curve. So in the case of our experiment this week, the known concentrate, we're going to be putting pieces of potato into known concentrations of sucrose and measuring their mass before they go in the sucrose versus their mass after they went in the sucrose to see if they gained mass or lost mass. The idea being, once again, let's go back to this slide, if they're put in really sugary water, we would expect the water in the plant cell to leave, causing them to get lighter. If they are put in very pure water, we might expect them to gain water and become heavier. So that's what we'll be measuring, and so our x-axis is going to be sucrose, or perhaps we'll use salt this week, depending on how we set up the lab, but we'll have sucrose solutions on one axis, and we will have change in mass along the y-axis. And then you can put in a best fit line, and you'll have an unknown solution, a solution where you don't know what the sugar concentration was, and you will use your graph to predict what that unknown concentration was. The way you'll do that is you'll say, oh, how much mass did I gain or lose? And you can draw a line straight over from that to the best fit line and straight down, and you'll get a really good idea of how concentrated that solution was that you placed your potato in. Okay? So if you don't quite understand that, Google standard curve and just read a few definitions about it, and you'll get a much clearer picture. It's okay to research things further if you don't quite understand them in the lab, and I highly encourage you to do so. That will make you a very successful student in the future. All right. Don't forget to take your pre-lab quiz, and I will see you very, very soon.